Prof from Queensland University of Technology, now a visiting professor at Oxford, will discuss exploring the sources and effects of variability and heterogeneity in cardiac electrophysiology modeling.
Okay, long title. So I'd like to thank the organisers very much. It's great to be in Cambridge, the other university. I'm afraid I didn't actually stay away from non-spatial things. There are two parts to this talk. The second part is some non-spatial work that I've done in heterogeneity. So I apologise for that in advance. And I usually forget to give my acknowledgements, so I'll do them now. So most of this work was done at Oxford. I've been at Oxford for 8 years in a shared position in the computational cardiac modelling group led by Blanca Rodriguez. So this is joint work with people in that group and PhD students and postdocs. Some clinical data from Szeged in Hungary and the John Ratcliffe Hospital in Oxford. Colleagues in Australia, including my wife Pamela, and some colleagues in Spain. Okay, so some overview, some issues in electrophysiology and how you cope with variability. So, and I distinguish between variability and stochasticity. Of course, there are subsets there. So we all know what a heart is. We all have one. It's got 4 chambers. Here they are. There are atrial chambers and ventricular chambers. And there's an electrical wave that propagates through the heart, and that it's called the action potential, and it causes the heart to beat. So it's electrophysical and also mechanical. I'm not doing anything mechanical here today. And the form of this action potential takes this sort of form as it goes through the atrial nodes, into the common bundle, through the ventricular cavity and through the along the fibers. So it gradually changes this shape. But this is the fundamental shape. You have a depolarization, a repolarization back to a steady state. And so myocytes, cardiac myocytes, are excitable cells. So they're excited by stimulus, which then propagates through the heart, causing the heart to, at this point, calcium kicks in, and so it's the calcium channeling that causes the heart to contract. And on the right-hand side, we just see, so here's the action potential again, and these represent currents through different channel types. So sodium, calcium, different types of calcium, different types of potassium, and so on. So embedded in the myocyte cells are different types of ion channels which open and close in certain ways, and are responsible for the ionic fluxes that go in and out of the cells. Okay, so as I said, the ion channels are embedded in the plasma membrane. Here's an image of, so the myocytes are lined in fibers that wrap around the myocardium and also arrayed in sheets. And here you see this would be from a healthy patient. You see the fibers are aligned in these lines. In between there are pores, there's collagen, there's fat, there's blood vessels, all embedded within the tissue. The mathematical models that are used are ODE models to describe the behavior of a single cell, a single myocyte. So here's a typical cartoon. Here's the membrane. Here are the ion channels embedded in the membrane, and fluxes take place. And then these ordering differential equations are coupled to partial differential equations, which are essentially reaction diffusion equations that describe how the action potential propagates through the tissue. So I apologise for some spatial things, but structure cardiac tissue is incredibly heterogeneous. So one of the things I will be talking about at the end is what does that mean for the sort of mathematical models that you may build to describe the propagation of an electrical wave through this tissue. But the first part of my talk is about differences between people, animals, so inter-subject anatomical variability. So this was a study done by a student at Oxford of Blanca Rodriguez. It's 4 rats. In the ventricular cavity, there are Purkinje fibers that hang in the cavity and accelerate the propagation of this wave through the tissue. And this is the network of hanging fibers. And these rats were all healthy at one point. Same age, same sex, but very, very different in terms of the structure of these Purkinje fibers. Yet the hearts functioned in the same way. So you might ask, well, why? And here, as in terms of the spatial aspects, there's a disease called hypertrophic cardiomyopathy. Sometimes you hear stories of young athletes dropping dead or having a heart attack on the football pitch. It's happened in the Premier League, for example, in the UK. And it's often because of this disease where the heart enlarges and the fibers that are normally smoothly wrapping around the myocardium become disarrayed and affects the propagation of the electrical wave. So this is a diseased patient, and here's healthy cardiac tissue. So again, if you want to build mathematical models and you want to do personalized medicine, how do you account for these different structures in your models? And finally, a little bit more about physiological variability, which will be now the first part of my talk. Here are ionic currents in 35 different human patients. You see that, this is just for a potassium, you see that they're very different. Here is the action potential in rabbit Purkinje fibers, so different rabbits. And here you see the wide variety, a wide range in the action potential. So the biomarker, one biomarker is the APD, it's the period of this wave. So here it's around about 200 microseconds, but it can go anywhere from 200 milliseconds to about 350. So you have all this variability between subjects.
And the same subject, how much variability is there? The same.
There's less, but there's still variability. There's still variability. And here are 2 patients, their ECGs, and again, they were both healthy, and you see they look different. So, a little bit about how you build mathematical models, the sorts of data we have. I don't want to go into this too much. There's ECG data, there's isolated patch clamp. So you take a single cell, you clamp it, you inject a current in it, and you can measure the response to the change of the current you inject into the cell. You can do optical mapping, so that the tissue will fluoresce. You can do in vivo experiments after surgery. We can get histologies. You can grow a heart in a dish. You can use differential tensor MRI, where you magnetize water molecules and you flip the orientation, and so you can track how the water molecules preferentially follow structures in tissue. So you have all these different data sources, and you want to calibrate your models against those data. So this is the sort of model that you build mathematically. So we have a cell model, so it's just an ordinary differential equation that relates voltage to a stimulus current, and this IM is the sum of ionic currents in and out of the cell that are described in some way. You might have seen Hodgkin-Huxley equations, for example. That's a particular example. So you have gating variables. So here we have sodium. This has 5 gates, 3 M gates, an H gate and a J gate. Each gate can open and close in terms of some ordinary differential equation, and these alphas depend on the voltage themselves. So this is a highly nonlinear problem, and then you have to propagate the voltage through the tissue described by some partial differential equations. So that's the fundamental model for modeling electrophysiology in the heart, or indeed in the brain. Okay, so that was the background. So a major problem in drug development is the issue with identifying those at risk of arrhythmia. And it turns out that many drugs that are developed have the possibility of being pro-arrhythmic for some classifications of populations, some age groups or whatever they might be. So in order to understand these effects, one needs to develop modelling formulations that accounts for the variability across populations. And how do you do that? So if we can do it, then we can have a very powerful mechanism for understanding variability at population level. So this is sort of partly where Omar was coming from. So the question is, how do we account for that variability at the population level? So this is not about developing stochastic models. It's about doing something called populations models that, for example, have been used in climate modelling for many years. So what is this population of models approach? So it's very simple. Let's think of it. You have a cell model described by an ordinary differential equation, has a certain formulation, has a number of parameters that maybe are unknown or characterizable to certain stratifications of population. So the idea is that you calibrate a population of models against the data you have. So you identify what your key parameters might be. In the case of these cell models, they're the conductances associated with the ion channels. And then you search over parameter space in some sensible way, and you ask the question, choose a point in parameter space, I run my model. Does then that simulation calibrate against the experimental data I have, whatever that experimental data might be? If it is, then that model is in my population. And you do that a number of times in some appropriate way in terms of sampling this parameter space, and you build up this population of models. And that population of models is a proxy for the behaviour of individual people, for example. And then you can do things with that populations of models in a probabilistic manner. You can ask questions, you can try and stratify that population into age groups or sex. You can ask the question, if I do drug block, I want to look at the effect of drug on a subset of populations, I can apply it to my population of models and see the response. So it's a very powerful probabilistic way of looking at variability between across populations, but also very simple. So this was originally proposed in the context of neuroscience by Yves Marder, who's at NYU, and then was adapted by the group at Oxford to look at it in terms of cardiac electrophysiology. So this would be a single action potential, and this would correspond to data that we have of action potentials across some population of people. And this would be a single simulation. And so any simulation that might occur in here, based on a certain parameter set, would go into the calibrated population of models. And if it occurred out here, of course, it wouldn't go in. And so you build a population of models in that way.
So you may have a lot of different variations in parameters that give you the same kind of variability in phenotype. How do you decide?
So it's a sampling problem. So I'll talk about that in a minute. That's a good question. And so it just means that you do your modelling in a different way now. It becomes a probabilistic approach rather than taking a single model with a single parameter set and just using that to try and understand the underlying variability. So populations models. So physiological variability is difficult to investigate with experimental methods. It's one of the biggest challenges in biology and medicine. Sex and age differences have been identified as important determinants of inter-subject variability. This populations and models approach was pioneered by Marder about 8 years ago in the context of neuroscience. And we believe they provide new insights into sources and implication of variability in cardiac electrophysiology, but in other settings as well. So in the context of cardiac electrophysiology, the sorts of data that you have come from this action potential. So what gets measured is the peak amplitude, the dome peak here, the velocity of the upstroke, the period of the wave, so this is called the APD, or you might have something called APD 90, where you take 90% of the value from here and you work out the period at the 90% level. So you measure a number of biomarkers and you measure that across a population. And then there's many ways of calibrating the model. One way is just to say, well, I have biomarker 1, and for my population of 100 patients, I have a range. So when I choose a point in parameter space and ask, does that model fit into the population or not, it just has to lie in the range of all the biomarker information I have. That's one way of doing the calibration. If you have time series data, you can provide additional calibration just by doing some mean square comparison with the time signal if you, and the simulation if you have it. If you have enough patients, you can do more sophisticated things. So you could look at, for a given biomarker, you could look at the distribution of values of the patients for that particular biomarker. And so when you build your population of models, when you calibrate your population of models, you would try and build it in such a way that it matches the underlying distribution in the data that you have for those biomarkers. But that only makes sense if you have a relatively large population, several hundred, for example. Okay, so somehow we have to sample parameter space. So that's the key. So building the populations is easy enough. How do you sample parameter space? And there are a number of ways of doing it. Some are reasonable, some are not so reasonable. So this is, I'll describe something called Latin hypercube sampling. So it's best described in 2 dimensions. So it works in an arbitrary number of dimensions. So here I have 2 parameters, P1 and P2. There's a certain range of values that they line from A to B and C to D. And I discretize my 2-dimensional parameter space under some grid. It need not be uniform, but here it is. And Latin hypercube sampling just says, I'm going to place a cross, but only can be a single cross in a row and column. So this would be a trial of 4 samples under Latin hypercube sampling. And what does the cross mean? The cross means that you choose a point at random in that little box. So when you're building a population of models, I generate my Latin hypercube sample. I choose a point at random in that box, there it is. I run the model, and then I see if it's calibrated against the biomarker data that I have. If it is, it's in the population. If it's not, it's not. So the point about Latin hypercube sampling is that it doesn't scale with dimension. So in 3 dimensions, you can think of this as you have a single cross in a plane, for example, in a row and a column. So it doesn't scale with dimension, but of course, you get a sparser number of crosses as the dimension increases. So some people ask the question, well, does that matter? How well should you cover parameter space when you build your population of models? And the answer to that is it depends on the questions you're answering. If you look at this, you see there's a gap here. So you might say, well, if I did that, I'm not very happy because I've got this gap here and I've got this gap here. So there's something called orthogonal sampling, which would require you to put one cross in each of these 2 by 2 blocks. So it's a slightly more uniform coverage of parameter space. And you can work out estimates of what sort of coverage you get, and I won't go into that because it's just a bit of math. But so you can estimate percentage coverages in terms of the gradation of the mesh and the dimension of the problem. Okay, so because a lot of people have asked this issue about building the population, how do you know if you covered parameter space in a reasonable manner or not? So what we did in this little study, and it was purely with synthetic data because we wanted to do a detailed comparison between Latin hypercube sampling and a SMC Bayesian approach. So a Markov chain Monte Carlo approach using a Metropolis Hastings algorithm, essentially. So we took a cardiac cell model, it's called the Beta-Reuter model. So it's 40 years old now, but it still produces a very nice action potential. So it's a very good model. It's just there are more sophisticated models now. So it's just a system of 8 ordinary differential equations that describes the excitability of a ventricular myocyte. There are much more sophisticated models now, but this is still a very good model. And so we chose that model so that we could compare our different sampling strategies in order to build our populations models. So we compared Latin hypercube sampling and a new algorithm, which is a sequentially Monte Carlo approximate Bayesian computation algorithm. And we compared it in the sense of asking what sort of distributions or models do we build under these different sampling techniques, and what is the effect if we then applied some out-of-sample prediction. So if you build your population of models, you can take that population and say, well, what's the effect of drug rock on that population of models? Thinking that this population of models now is a proxy for people. So then you apply some drug block protocols to that population of models, and you see what you see. And so you ask the question, is what do you see? Is it affected by the way that you did the sampling in the first place? So those are the sorts of questions that we just wanted to look at. So this SMC approach is essentially, you think of points in parameter space as little balls, and essentially you're moving these balls around, you're diffusing these balls around in some way based on the underlying distribution that is described by the data you have. And this iterative algorithm will settle down on some probability density that describes the population model's structure. It's a good test model because there are essentially only 4 parameters that make any sense, though the maximal conductance is associated with the ion channels in this model. So we can do a detailed study and compare the different sampling approaches. How am I going for time? 15 minutes or 20 minutes? So this is what we found. So first of all, this SMC approach is much more efficient than Latin hypercube sampling. This SMC approach, you start off with a number of balls representing your parameter points, and you move these balls around in some way, but you don't throw the balls away. You just keep on moving them until you find that they lie in some appropriate distribution. Whereas with Latin hypercube sampling, you can imagine that you have to do many, many trials to build up a relatively large population of models. So SMC approach will be more efficient than Latin hypercube sampling. But the question is, does it give you the same sorts of distributions of the populations and models? So here you see, this is just against two conductances. SMC is red and LHS is blue, and just random sampling was yellow. And you can see that there are some differences in the distributions. So, and again, you can see it over here. So you see LHS and SMC. So the underlying distributions that you build, the blue ones, are a bit different. Not a lot different, but a bit. And the red ones here correspond to when you do drug block, and you compare them. So the take-home message for this little study is that if you want to build a populations of models, Latin hypercube sampling is a very simple, parallelizable approach. It doesn't matter that much if you don't cover large sections of parameter space, because it's not, what we're doing is not parameter fitting, we're exploring variability, and it's a different question. On the other hand, there are some differences between doing this Bayesian approach and LHS. So you get some sorts of slight differences in the underlying distributions that you build with your populations. And the reason is because the SMC Bayesian narrows in on the underlying distribution, whereas LHS is you just choose points at random, essentially, under the sampling rule. So in some sense, there's greater intrinsic variability in the Latin hypercube sampling approach than in this SMC Bayesian approach. But it's subtle, and they're fairly similar. So, well, I don't read all that, I've more or less said it. Just to say that although this was a synthetic study just to compare these approaches, we've applied these ideas in a number of settings where we've looked at things like characterizing alternans. Alternans are just where you have an action potential, and rather having more or less the same APD, the same period, the period can alternate between quite two different values. So it's like a period 2 oscillator in some sense. So we've applied these ideas to looking at variability in a number of settings, and that was one of them. Okay, so that's the first part of the talk. So nothing conceptually difficult, except to say that we go away from this traditional approach of a single model with a single point in parameter space. And that's well and fine. That's perfectly fine thing to do. But if you want to understand variability at the population level, you have to do something different. And so this population of models approach is doing precisely that. And it's very similar to what climate models have been doing for many years. Okay, so the second part, and I'll skip through this fairly quickly. It's spatial, so people who don't like spatial stochastic things can ignore this.
It's going to be okay.
So this is a dense slide. What is it? So, as I said before, if you're trying to describe the propagation of this electrical wave in tissue through the heart, essentially you have an ordinary differential equation that describes the behavior of a single myocyte, and that's coupled to a partial differential equation, which is essentially a reaction diffusion equation that describes the propagation of the wave. Call it a cable equation, if you like. And so that's the coupling. So you have this reaction diffusion equation. This ion is the sum of the ionic currents, and it's that term there that couples to the ordinary differential equation for the cell model. And that's the thing you have to solve through time. So what we've done in this study, so if this is constant, you can take it outside, you have a Laplacian. We've done something that looks silly. We've replaced the Laplacian here in this equation by a fractional Laplacian. So instead of having del squared u del x squared plus del squared u del y squared, we have that thing but raised to the power alpha upon 2. If alpha is 2, you get the standard Laplacian, and if alpha is a number between 1 and 2, you have a fractional spatial derivative. And then you might ask, well, why would you do that? So as soon as you do that, you have something called a non-local operator. And so our initial instinct in all of this was that non-local operators would better capture the spatial heterogeneity of cardiac tissue in terms of the propagation of this electrical signal. So that was our instinct, and then what we've attempted to do is calibrate against data just to see how well that idea fits. So very briefly, here's the cable equation. It's coupled to an ODE. It produces an action potential that propagates through the tissue. It's pretty simple. Heart's a pretty simple thing. That's all there is to it. So what we've done is essentially replace this Laplacian here, Laplacian of the voltage, by a fractional Laplacian. That's all we've done, nothing else. And so the question is, if we do that, can we recover important phenomena of cardiac tissue by considering this different formulation of wavefront propagation?
This fractional Laplacian has non-local properties.
Yes.
Why did you not use just a kernel?
Yeah, so yes, you could use some appropriate kernel, but in fact,
Which has already non-local effects.
yeah, you could do that. But in fact, why not do this? Is a different question.
Why?
No, but you could do. So I come back to that in a minute. All right. So there's a reason for why I've done this.
Am I remembering correctly, the fractional Laplacian is really just the operator that goes with the stable process?
With the what, sorry?
With the stable process?
Yes, you're exactly right. You're exactly right. So you're exactly right. So you can view this equation, well, forget about this, as being the probability density function of an ensemble of particles undergoing a certain stochastic process. And the stochastic process is an alpha stable Levy process. So you can view it, so that's where the stochasticity comes in. Okay. And that Levy process is a jump process. I'm not saying ions jump, so that's not what I'm saying, although some people think I do. Yeah, so here we go. But I have a different interpretation of why this makes sense. And it's to do with potential theory. So in actual fact, you can define a fractional Laplacian operating on some function just in terms of some appropriate integral. You can take the inverse of that thing and you get some other different integral. You put n equals 3, and if you take the value of alpha equals 2, then this phi would behave like 1 upon r, which is like a monopole. And if you put alpha equals 1, then this phi will behave like 1 upon r squared, which is a dipole. And if you take an alpha between 1 and 2, you get some intermediate properties between a monopole and a dipole. So what we say is that all of the spatial heterogeneities in the cardiac tissue, they create secondary sources of electrical fields. So this difference here will create a secondary electrical field. And if you add up all those secondary electrical fields, you'll get some behavior that's between 1 upon r and 1 upon r squared. So that's the reasoning behind why we use these fractional differential equations. Now, one could maybe do this in a kernel setting, but it wouldn't be quite so clear to me how to do that. So all we've done, and I'll go through this very quickly, is we've asked the question, well, can this explain some properties that traditional models cannot explain? So this first experiment was heart cells in a dish. So there was a group in the States who plated down neonatal rat cells in a dish. and they can control the orientation of the way these rat cells connect to one another. They're mycete cells. And you can stimulate the cells, so you apply some stimulus at the centre, and you can measure the action potential as it propagates through the dish. And what you see is this shortening here. So at the centre, the action potential looks like that, and near the boundaries it shortens. So you can do the same thing with your mathematical models, and with the standard formulation, you don't see the shortening of the APD. All right, so you might say, well, I'll change the underlying cell model in some way so that you can see it. And it's true that if you change the cell model in certain ways, then perhaps you could see some shortening. But as soon as you apply a non-local model in the right way, you immediately see a shortening in the action potential, and it looks very close to the experimental results. So that's one piece of information. Second piece of information is, this is clinical data from Ben Hanson's lab in London, where you look at the APD, so the period of the action potential wave, and you plot it as a function of the activation time. And this decay is thought to be a very important property in healthy hearts. So what you can do is you can take your standard reaction diffusion equation or the fractional diffusion equation, coupled to a cell model, run it in one dimension only, and see what you see. And again, in the red is the standard diffusion approach, and again there is no shortening. You don't see this shortening, which is absolutely vital in healthy hearts. In the case of the fractional model, as you vary the alpha, again you see the shortening. So again, that's sort of interesting. And then finally, what we did was a DT-MRI study. So I won't go into this slide in too much detail, just to say that in DT-MRI, you make an assumption about the diffusion of protons under an external magnetic field, and you would make the assumption that it's essentially Gaussian diffusion, that it's normal it's diffusion under a normal distribution. So that leads to a so-called exponential decay term that arises. If you postulate that the underlying processes are based on some fractional differential equation, you have to change your model in some way. So what this represents is a slice of tissue in rat, and the ADC is a map of the average eigenvalues associated with the diffusion tensor that you measure when you do DT-MRI. So ADC is just the sum of the eigenvalues of the diffusion tensor that you calculate. Fractional anisotropy is a different measure. We've introduced a new measure which is called the apparent anomalous coefficient, which is the sum, which is the average of the fractional powers. So if all the betas were 1, this thing would be 1. So the further this thing is away from 1, the more the influence of the fractional derivatives come into play. And already you can see that. You can see that in the data here. And I've nearly finished. So what we did was we did a small study of six patients. 3 were healthy. There are lots in the John Ratcliffe Hospital. 3 were healthy, 3 were diseased. They had hypertrophic cardiomyopathy. And we did this DT-MRI study, and we looked at fitting a standard model and a fractional model to the data. And what we were trying to do is estimate the fractional exponents in each of the 3 dimensions. And so here, the AAC is the key one, so nearly everything is red, so that corresponds to the values being 1. So that would be standard diffusion. That's in systole. It's when there's ejection of the blood from the heart. In diastole, you see some differences, but whether you can say that's evidence of the fractional model making sense is debatable. But in the disease patients, we find something that's interesting. So here, again, this is a single slice of the tissue. And what you see is regions where the average value is far from 1. Remember, if they were all 1, that would correspond to standard diffusion. And here, in these blue regions here, the average value is far from 1. And not only that, you see this heavy tail that you would expect with a non-local behaviour. So I've skipped over a lot of details, but what this fractional approach seems to give us is that we have a way of constructing a spatial biomarker in a cardiac setting, the spatial biomarker being the fractional exponent in our operator. And we believe, although we need much more study and many more patients, that could be a way of characterising different types of diseases that hitherto were very hard to characterise. Okay, so I've finished. So just some final thoughts. These first two, these points 2 and 3, because I described to you in the second part of the talk something about non-local operators, I probably didn't do a very good job. They're not intuitive. Ions don't jump. So does that matter? Well, it matters that ions don't jump. But the issue of should a model be intuitive or not, does that matter? On the other hand, if we're looking to come to a regime of personalised medicine, there needs to be some trust in the community about models that are used. So perhaps it does matter. How do we capture the different levels of variability? So the approach I described is via this population of models, which acts as a proxy for the variation in a population of people in terms of their biomarker responses. In doing this, I've had to learn a lot of things that I'd never studied as a student, especially Bayesian statistics, which I sort of stayed away from when I was young. Of course, I didn't understand it, I guess. But I've now come to the conclusion that I shouldn't do that. And so I think there's a greater linking now between the modelling and the Bayesian statistics under this sort of population of models approach. And now we just started a new study where we're actually using real data, where we're comparing 3 atrial models in a population of models sense. So here you have 3 models. So they give slightly different behaviours. So how can you understand the behaviour of these models? If you build populations of models and then compare the underlying distributions, then I think it starts to tell you more about the models themselves, and it certainly tells you a lot about the underlying physiological variability. Okay, thank you. I'll just drop my glasses. Sorry. What was the one? Yeah. Yes, sir.
You have this population of models for healthy people, but you can do it even for whatever, right? I mean, if you have done is there any, the distribution, do the distribution overlap when you do this variability for healthy people and unhealthy people?
Yes, they do. The distributions can overlap, yes, they can. You would expect them to do so. But it's, it is a way, I think it is a way of understanding the responses of different sections of a population in terms of age and sex and race and many other things, which you can't really do with the standard modelling approach. Yes, but you do see overlaps, and you would expect that.
Thank you. So for the Bayesian part that you were talking about, it wasn't very clear to me. What are you using for your priors, or are you constraining your prior probabilities at all for the parameter values?
Yeah, so we're moving the particles basically using a Metropolis Hastings algorithm, and we make an assumption that there's some underlying normal distribution with some covariate function, and we're moving it based around that
And you
covariate
sample
function.
from
Yes,
that.
yes, yeah. I mean, there's other things one could do as well, but
Yeah.
this seems to work.
And
well.
when you did the light and hypercube sampling and the other type of sampling, a lot of people do this Sobel Halton sequences mapping the
Yeah.
hypercube. Is that of any use for you guys?
I did look at it, but I didn't find any. Yeah, there's a whole space of this
Yeah.
combinatorics and discrete mathematics. I didn't use it. I did look at it, but I couldn't find a reasonable use for it,
Okay.
so I didn't use it.
Fair enough.
Can I ask Vikram to come up? Instead setting up.
So I didn't quite understood. So you in your Latin hypercube sampling, you also combine that to ABC, to approximate Bayesian computation?
No, not in that case. No, we don't do that. So,
You sample a parameter set with the Latin hypercube sampling,
so we're comparing two different approaches. One is the ABC.
but you still need to compare the model output with the data. Do you do it in a approximate Bayesian computation way? You just calculate the distance.
Yes, exactly. Yes, exactly.
And what was the conclusion of it? Like, exactly, like I don't know. You think that hypercube sampling is as good or is not as good?
So the conclusion is, it depends what does good mean. The conclusion is that both are reasonable approaches for capturing the underlying variability. The SMC is more efficient, but in some sense it tends to really concentrate down on the underlying distribution of models, whereas the Latin hypercube samples in a more general way, and so is perhaps it's slightly better at capturing the underlying variability of whatever you're measuring.
I have a question. So the utility of population models approach, I think, is clear for physiological scales. Would you suggest that one should take this approach for cellular scale models? And if so, then how do we distinguish between inherent stochasticity
Well, yes.
and variability at that level?
Yeah, that's a good question. I mean, so in the case of these cell models, you can put stochasticity in the ion channel level, but the stochastic effects are fairly small. Otherwise, we'd all be dropping dead. But there is some stochastic variation, but it's fairly limited. Whereas in the sort of settings you're talking about, that's certainly not the case. So somehow you have to distinguish between both of those things.
Any idea how?
I'd have to think about that. But I think there's ways of doing it. I should say that Pamela's done some work where she's applied Latin hypercube sampling just to stochastic differential equations. And you can calibrate your models. You run 20 samples. If each of the samples lies in range, then that's in your population, for example.
Let's thank Dr. Burrage.